DNA then is now going to be replicated. That's where we're going in this video. And let's start with prokaryotes. So here's E. coli. Now prokaryotes don't have a nucleus. They have a single ring of double helix DNA. I'll just draw two circles here. That represents the double helix of DNA. And this is called a nucleoid. So that is the single chromosome of a prokaryote. And it's not inside of a nucleus. It's just in the cytosol, in the cytoplasm there. Now there's an area where enzymes attach called the origin of replication. That's where division is going to begin. And division begins there and goes left and it goes right. So we can see in this next drawing, we can see the daughter DNA, that's the new DNA being produced. And we can see the original parental DNA opened up there. And so it's being copied in both directions. Basically, it's copied all the way around until the enzymes meet up at the bottom there, and we have two new strands of DNA produced. So there's a bit of terminology here. You can see in this drawing, we can see that this entire thing right here is known as a replication bubble. And then 1/2 of a replication bubble, which kind of looks like this. I'm just going to draw it really crudely, right? So there's half. If there was another side over here, this would be a bubble where it's all opened up. But we're only looking at half of it. So this is called a replication fork. Just half of the replication bubble is the replication fork. You can hear my kid playing in the background, but that's just the nature of teaching from home. Okay, the DNA continues to copy itself until you have two new double helices made, two new nucleoids made. So at the end of this, we have two of these nucleoids produced. Both of them are double helices of DNA, and those can go off into two different bacteria. Now, in eukaryotes like us, we don't have rings of DNA. We have chromosomes. Chromosomes are linear pieces of DNA. So they are double helices of DNA that are linear. And so we have multiple origins of replication along the length of our DNA. DNA is copied from left and right in both directions until our replication bubbles run into each other. And then we have, then they separate out into two new DNA molecules. There are some videos that you can watch that will help to make that more clear. But for us, we have a lot more machinery going on because we can't just attach a couple of replicating molecules and go in a circle. We have to go left and right along this linear DNA strand, and that's how that's going to work. Now, some of the major enzymes involved in this process. One of them, this one right here, is helicase. Helicase is like a zipper pull. It attaches to DNA and it breaks the hydrogen bonds, opening up the parental strand or the template strand of DNA. Okay? And it does so from a 5' towards a 3' direction. There's also a protein called single-stranded binding protein. I'm not going to get too worried about it, but I do want you to at least be familiar with it. What it does is it prevents the DNA from closing back together again before the cell is ready. So it prevents the template strand from closing again and reforming those hydrogen bonds. Topoisomerase, that's another one I'm not going to get too into, but I'll just mention it here. Topoisomerase's main job is to prevent the DNA from getting all tangled up or broken. Imagine trying to play with a long string and you're trying to do something down here and over here it's swirling around and getting into a knot. At any time I play with string or electrical cord, you know, like trying to wrap it up and put it away, it makes a big knot. So topoisomerase's job is to help prevent that. Prevent DNA from getting all tangled up or broken while it's being copied. It relieves tension. And primase, it's got a job that we'll talk about in a little bit. It creates a little bit of RNA, something known as a primer. And you can read this more later. We can come back to it. But that's one of the other major enzymes that we're going to talk about soon. There are other enzymes as well, including these two, which are, we can just call DNA polymerase. But there's DNA polymerase 3 and DNA polymerase 1. Those are some of the major molecules that make new pieces of DNA. So and we'll talk about a few of those as we continue. So let's look at a replication bubble. There's a lot going on there, and I want to show you that, first of all, here's the origin of replication. Enzymes attached here and here. The first thing that's going to happen is helicase opens up that DNA. Okay, helicase opens the DNA up. So helicase would be like right here in this drawing. And there would be another one right here in this drawing, because this is a replication bubble, right? So helicase is there, and it's moving that direction, and it's moving in that direction, those two. So the first thing that's going to happen is we're going to start copying that. After we open this up, we're going to attach the enzymes, and we're going to begin to copy this piece of DNA, and we're going to copy this piece of DNA down here. Now, things to look at overall. There's something called the leading strand and the lagging strand. Just look at how they're drawn differently. The leading strand is one big piece. The lagging strand is shown in little chunks. I'll try to find some videos that help to explain that. This is probably a lot easier to teach in person for me than online, but I'm going to do my best to explain it to you here on this PowerPoint in the next video or two. So the easiest one to explain is the leading strand. Now the leading strand is made in one continuous piece. So what happens first is DNA helicase attaches, and it unzips the DNA. It opens up the replication bubble or the replication fork. Then what happens is, single strand and binding protein sticks on there to prevent the DNA from shutting. Okay. The next enzyme that comes along is going to be primase. Primase builds this RNA primer that we see right here, this red thing right here. That's the RNA primer. That RNA primer is only a few bases long. It's not very big. So first thing that happens, DNA unzips. Okay. The next major thing that happens is primase comes in, and it's going to build a short primer. If you don't know the word primer, when you need to pump an old-fashioned well, you pour water into it first, and that primes the pump, allowing it to pull water out of the ground. Or if you've ever shot a shotgun, the primer is at the end of the shotgun shell. It sets off the shell when the firing pin hits the primer. A primer is something that initiates the reaction, okay? So the primer is necessary for DNA to be replicated. So without the primer, DNA polymerase, the enzyme that makes new DNA, cannot attach to the template strand, okay? It can't attach and make a new copy. So the DNA primer is built, and then DNA polymerase, which is what this is DNA polymerase 3, attaches to the primer, and it makes a copy of that DNA from a 5' to 3' direction, meaning the new piece of DNA is being built by adding on to its 3' end over and over and over again. And that DNA polymerase, as you can see in that picture, is following helicase. It's following helicase and making a new copy of the leading strand. And it does that all the way until it runs in to the next replication bubble, and then everything comes apart and it disassociates. So just to show you that in this drawing here, here is the origin of replication. We attached a primer, so we made a primase attached and made a primer. DNA polymerase 3 attached, and it followed helicase, and it made a new copy of DNA in this direction, and it would still be continuing in this direction as helicase continues off that way. Same thing over here. Here's one, and it continued off, and helicase is moving in this direction. So DNA polymerase 3 would be right here, following helicase. Okay. In the next video, we're going to talk about the lagging strand. So I will post some additional videos in Blackboard explaining this, showing how DNA is replicated. Please watch them. This is a very visual process. I think it will benefit you to see those videos.